with the presence of antibodies. It just says you've been exposed to some Mycobacterium sometime. The cytokine assays that are coming out, there was big hoo-ha about that in the diagnosis, but in the field they don't work very well to distinguish active disease from latent infection or early exposure. So the cytokine assays are turning out to be of limited use as well, and are actually registered for use as a diagnostic aid and not as a diagnostic. And the skin test is useful only in our unvaccinated population. That really only happens in the United States. And it doesn't happen in our immigrants who come in to be citizens. They're all vaccinated because 80% of the world is vaccinated at birth. So our diagnostic techniques are as bad as your diagnostic techniques, and most of the time we do chest x-rays and make the assumption if someone's had a cough for 2 weeks and their chest x-ray looks bad that they are suspected tuberculosis. This is a great picture in Ethiopia. I was visiting a hospital in Hosanna, and they brought me to show me their very wonderful x-ray machine, which they had gotten 2 years before. And as we were all standing in the room admiring this lovely x-ray machine, they mentioned that they were hoping to get enough money to buy the lead shields in the next year or two, at which case every male in the room leaped out and was gone from the room and gone from the hospital. So, yeah, so we don't do particularly well in TB for diagnosis either. So if you think you have trouble with MAP, you're not alone. So let's talk about acid-fast bacteria. There's always this issue of is MAP acid-fast or not in vivo? Our latent TB problem, and we have there are 2 billion people in the world out of 6 billion that are running around with Mycobacterium someplace in their body. We have no idea where it is. We can't find it. We can pretend like we know what latent TB is, but we don't. If we cure a mouse with drugs, they will be free of any disease symptoms for a good 6 months, and you can grind that mouse up and plate the entire mouse, and you will not find a bacteria that will grow out. But if you leave the mouse for 6 months, he'll come down with disease and die of TB. So they're there. They're someplace. We don't know where they are. They're thought to be intracellular, but hey, what do we know? We don't find them inside the cell, we don't find them inside the tissue, and we don't find them inside an organ. So the other thing I should tell you is that certain drug-treated Mycobacterium tuberculosis become acid-fast negative, although the drug is ineffective for killing them. So there's many ways for an acid-fast bacteria to lose its acid-fastness, and that doesn't really reflect on anything other than the fact that the bacteria is adapting to something it doesn't like. We have no idea in TB whether our latent bacteria are oxygen deprived or nutrient deprived. We make the assumption that they must be because they don't seem to be replicating, but we don't exactly know what's preventing them from replicating or whether they're doing it themselves as a survival mechanism. So when we talk about latent TB, in our studies, we basically do everything to them we can to make them stop replicating, and then we say they're latent. But it's a joke. So what does it take to become a public health hazard? So there are about 850,000 people in North America with Crohn's disease, and 50% of them are children and young adults. The non-2 CART15 gene abnormalities are detected in those studies that have been done in about 25% of Crohn's disease patients. We know now from Marcel's very nice work that non-2 regulates responses to neuronal dipeptide, and that mycobacterial neuronal dipeptide is actually 10 times more active than other bacterial NDP in stimulating non-2. And we know that MAP, which is, by the way, just a mycobacterium, can be isolated from a subset of Crohn's disease patients. So if we say that a biomarker for susceptibility to Crohn's disease from MAP is NOD2, then we have a potential 215,000 cases in North America that are putative MAP-associated CD patients, and does 215,000 cases warrant a public health threat designation? Well, if you look at all of the current diseases which cause hysteria in the public health systems, not just of the United States and Canada, but of the world, of course, tuberculosis, malaria, diarrheal diseases, AIDS, measles, hepatitis. But down here at the bottom was SARS. SARS infected 5,000 people and killed less than 500 people. And it was considered a major public health threat. I think the biggest issue here is that most of these diseases, but not all, are transmitted human to human. And so we worry about those things that can start as a nidus in one place and move from human to human out here. We haven't paid a whole lot of attention to those things that start in the cow and move into the human population, but move into vast numbers of the human population. And maybe we should start paying attention to that. What we need for determining whether Crohn's disease is in fact a public health problem is a sensitive, specific, non-invasive diagnostic or a biomarker that would help us figure out who has MAP infection and who doesn't. And until we do that, it's going to be very difficult for us to argue that this is something that needs to be paid attention to by CDC and the other regulatory authorities, public health authorities in your own countries. What we also need is an appropriate design of the appropriate antibiotic clinical trials. If only 25% of your patients have MAP infection that led to Crohn's disease, and a portion of those people, because not everybody responds to every therapeutic you give them, only a portion will respond to your antibiotic, you'll never get to a clinical endpoint for your clinical trial with that small population that can respond. So we need to figure out how to segregate MAP-infected people from the not MAP-infected people. Then we need to use antibiotics that we know work against MAP. And we know that they work not just in vitro, but when you think about TB and you think about MAP, remember that they live in little packets inside of a cell. So any antibiotic that works needs to go through the digestive system, get to the organ where the bug is, crawl into a cell, move to the phagolysosome, get in there in sufficient quantity to get into the bacteria, and then hit its target. That is a huge process for an antibiotic, and that's why we don't have just tons of antibiotics that work against TB. It's a huge task that you ask any antibiotic to do for an intracellular pathogen. So we need to, Michael Collins has been looking through a number of antibiotics that could work against MAP in vitro. Next step is in macrophages or other cell types that MAP can infect. And following that is in animals. And as Mike and I were talking yesterday, I have an interesting drug that works on all mycobacteria, including MAP. But if you were to do a cow clinical trial to tell me that it actually got to the organ to actually fit, I would have to pass him buckets of this antibiotic. So we need to find a smaller indicator system. Perhaps some of the mouse models that have been worked on at Marcel's group or others will be sufficient just to tell us that you can put an antibiotic into the mouth of a mouse and it will get to the intestine and it will go into the cell and it will get to the bacteria and it will kill it. But we need to think about ways in which we can determine whether your antibiotic works first before you do it empirically in humans. I remember talking to someone at Pharmacy in Upjohn about the clinical trial going on with anti-tubercular drugs. And his point was, well, if it doesn't work against, if they're not effective, then it's not a mycobacteria in CD. I was like, well, yeah, but they don't work against the bug in vitro. So why would you expect the 4 drugs that work on TB to work on MAP in vivo?